This procedure relies on plants expressing GFP in specific cell types, which can then be isolated from the rest of the plant cells using Fluorescence Activated Cell Sorting, or FACS. This example uses two marker lines expressing in specific cell types of the Arabidopsis thaliana root. Seedlings are grown in phytotrays or on agar plates. Their roots are harvested and treated with cell wall digesting enzymes. After 1 hour, the solution is filtered to remove large debris. The solution is then centrifuged and the supernatant is removed. GFP-positive protoplasts are subsequently separated by FACS. The sorted material can be used for cell type-specific analysis, such as genome-wide transcriptional profiles.
Hi, I'm Bastiaan Bargmann from the laboratory of Ken Birnbaum in the Department of Biology at New York University. Today we will show you a procedure for fluorescence activated cell sorting of plant protoplasts. In this example, we will be sorting two specific cell types in the Arabidopsis root. We use this procedure in our laboratory to study cell type specific transcriptional profiles. So let's get started.
To begin this procedure, grow wild-type seedlings as well as seedlings that have cell type-specific expression of GFP. The scarecrow promoter driving GFP marks the root endodermis and the quiescent center in one group of seedlings, and in another group, the root quiescent center is marked by the WOX5 promoter driving GFP. The seedlings are grown hydroponically in phytotrays on top of a nylon filter, which lets the roots grow through into the growth medium. Alternatively, plants can be grown on top of a nylon mesh.